Hello students, welcome to the lecture on genetics, and after this lecture we will be able to learn the following objectives. Describe the Mendel and inheritance, discuss to chromosome theory of inheritance, explain about sex determination in human beings, describe about linkage and crossing over. Let us begin with a brief introduction to genetics. Genetics is the study of how genes bring about characteristics or traits in living things and how those characteristics are inherited. Genes are portions of DNA molecules that determine characteristics of living things. Through the processes of meiosis and reproduction, genes are transmitted from one generation to the next. The Augustinian monk Gregor Mendel developed the science of genetics. Mendel performed his experiments in the 1860s and 1870s, but the scientific community did not accept his work until early in the 20th century. Because the principles established by Mendel form the basis for genetics, the science is often referred to as Mendelian genetics. It is also called classical genetics to distinguish it from another branch of biology known as molecular genetics. Mendel believed that factors pass from parents to their offspring, but he did not know of the existence of DNA. Diploid cells have a double set of chromosomes, one from each parent. For example, human cells have a double set of chromosomes consisting of 23 pairs or a total of 46 chromosomes. In a diploid cell, there are two genes for each characteristic. In preparation for sexual reproduction, the diploid number of chromosomes is reduced to a haploid number. That is, diploid cells are reduced to cells that have a single set of chromosomes. These haploid cells are gametes or sex cells, and they are formed through meiosis. When gametes come together in sexual reproduction, the diploid condition is re-established. The different forms of a gene are called alleles. In humans, for instance, there are two alleles for earlobe construction. One allele is for earlobes that are attached, while the other allele is for earlobes that hang free. The type of earlobe a person has is determined by the alleles inherited from the parents. The set of all genes that specify an organism's traits is known as the organism's genome. The genome for a human cell consists of about 1 lakh genes. Heterozygous and homozygous condition of alleles in organisms. The two paired alleles in an organism's genotype may be identical or they may be different. An organism's condition is said to be homozygous when two identical alleles are present for a particular characteristic. In contrast, the condition is said to be heterozygous when two different alleles are present for a particular characteristic. In a homozygous individual, the alleles express themselves. In a heterozygous individual, the alleles may interact with one another, and in many cases, only one allele is expressed. When one allele expresses itself and the other does not, the one expressing itself is the dominant allele. The overshadowed allele is the recessive allele. Let us now talk about Mendel and the laws of inheritance. Gregor Mendel was one of the earliest people to systematically study genetics. He devised two basic laws of inheritance. The law of segregation states that when an individual produces gametes, each gamete has only one of the two possible alleles from each parent. That is, the pairs of chromosomes 2n from each parent separate into two different gametes. One set of parental chromosomes per gamete, 1n. We now know that this happens during meiosis. The law of independent assortment states that different traits are inherited independently of one another. All the maternal chromosomes or all the paternal chromosomes do not end up in the same gamete, but rather the maternal and paternal pairs assort, separate into gametes independently of each other. The exception to the law of independent assortment is linkage. When two genes or DNA segments are located near each other on the same chromosome, they are more likely to be inherited together. These linkages can also be shuffled, however, by recombination, where the DNA is actually broken apart and rejoined. Linkage and recombination are very useful for scientists trying to determine the chromosomal location of specific genes or DNA segments, called mapping, as there is a direct statistical relationship between how far apart DNA segments are and how often they recombine. The birth of a science. Charles Darwin first conceived the idea of hereditary units when he published his theory of pangenesis in 1868. In this model, circulating units called gem mules are accumulated in the gonads and transmitted to the offspring. This theory was discredited by experimental tests performed by Francis Galton in the 1870s. Galton used blood transfusions in rabbits to show that the alleged gem mules in one rabbit's blood did not alter the heredity of the recipient rabbit's blood. In the 1890s, Hugo de Vries took the term pangenesis and trimmed it to pan-gene for the assumed units of inheritance. He argued that pan-genes remained inside the cell and did not migrate. It was this theory of intracellular pangenesis that led de Vries to independently find what Gregor Mendel had discovered 30 years earlier in his work with contrasting traits in garden peas. There are units of inheritance that are transmitted by reproduction. Wilhelm Johansen introduced the term gene to replace several contending and misleading terms for the basic unit of heredity in 1909. The term genetics came earlier when William Bateson coined the word in 1906 to represent the new field that studied heredity, variation, and evolution. Mendel identified what he called factors, later called genes, as the underlying cause for the appearance of certain traits in peas. He described them as stable units that seemed to disappear in a hybrid, but would reappear among some of the progeny of such hybrids. Mendel identified two laws, the law of segregation and the law of independent assortment, which together governed the movement of factors from parent to progeny. This strongly suggested that the factors of inheritance were discrete physical objects. Shortly before Mendel's work was being rediscovered, advancements in the construction of microscopes had allowed scientists to make careful observations of cell division. This led to the discovery of colored bodies in the cell nucleus that appeared to double and divide just before each division. These were called chromosomes. X-linked inheritance in hybrids. Beginning in 1907, Thomas Hunt Morgan extended Sutton's insights by conducting laboratory studies of the fruit fly Drosophila melanogaster. With his students Alfred Henry Sturtevant, Calvin Blackman Bridges, and Hermann Joseph Muller, he established what is now called classical genetics. Morgan and his students found new phenomena that added to Sutton's chromosome theory of heredity. The first finding, achieved in 1920, was X-linked inheritance, in which white-eyed flies showed a sex-linked inheritance of the trait in a modified 3:1 ratio. In other words, crossbreeding hybrid red-eyed flies resulted in all the female offspring having red eyes, whereas half the male offspring had white eyes. Morgan's team explained this modified ratio by proposing that the eye-color genes are carried on the X chromosome, of which females have 2 but males have only 1. This discovery further strengthened the case in favor of the chromosomal theory of inheritance. The male and female individuals possess different structural attributes in terms of chromosomes to decide the sex of the newborn. Genetic determination of sex is based on the chromosomes deciding the sex of the offspring. In these individuals, the chromosomes are of two categories: autosomes and sex chromosomes. Autonomies are responsible for all the somatic characters, while the sex chromosomes decide the sex besides carrying genes for some somatic characters also. In sexually dimorphic individuals, there is a pair of chromosomes meant to govern the sex. These are called X and Y or Z and W chromosomes. Structure of the sex chromosome. The X and Y sex chromosomes exhibit certain structural differences as revealed by cytological studies. In most of the organisms, the X chromosome is straight, rod-like and larger than the Y chromosome. The Y chromosome is smaller in size with one end slightly hooked or curved as in Drosophila. In man, however, no such curve is found in the Y chromosome. X chromosomes have a large achromatic area and hence carry many sex-linked somatic characters. The Y chromosome has less of achromatic region and carries but little sex-linked somatic characters. In dioiciously organisms, chromosomal sex determination is of basically two categories: heterogametic males and heterogametic females. XX, XY type. In man, Drosophila etc., where male are the heterogametic sex, the male and female have the following types of sex chromosome. This type was first studied in milkweed bug, Ligeus turcicus, hence it is also called as the Ligeus type. In the XXXY type, the male produces two types of sperms. 50% sperms would carry X chromosome and the rest carry Y chromosome. All the eggs are of only type. In all of them carry a single X chromosome. The sex of the newborn depends on the type of sperm carrying X or Y fertilizing the eggs. Linkage is a term used to describe the physical association of genes located on the same chromosome. When genes are located on the same chromosome, they do not assort independently, and as a result, linked genes are separated only by breakage or crossover during meiosis. When genes are closely linked, the alleles inherited from one parent tend to stay together. Correspondingly, the alleles from the other parent also stay together during assortment. The characters and associated with linked genes fail to follow expected Mendelian mathematical distributions in offspring when compared to alleles that are inherited independently of one another. Because linkage occurs on the same chromosome, one chromosome is one linkage group. When crossover occurs during meiosis, the linkage breaks down. Crossing over is the exchange of genes by chromosomes during meiosis that alters the genetic information contained within the chromosome. The further apart the alleles are on the chromosome, the more likely a crossover event will occur between their respective locations. The video here will show us the inheritance pattern of hemophilia.
Hemophilia is almost always inherited.
It is passed down in the genes, more specifically the X chromosome. Both men and women have 23 pairs of chromosomes. On the 23rd chromosome, women have 2 X chromosomes, while men have only 1 X and 1 Y chromosome. Since males have just 1 X chromosome, it only requires 1 defective X chromosome for him to acquire hemophilia. Females, on the other hand, have the advantage of possessing 2 X chromosomes. Therefore, both X chromosomes must contain the hemophilia gene in order for a female to become hemophiliac. If only one is defective, the healthy chromosome overpowers the defective one and leads to her simply becoming a carrier. When a gene exists but does not appear as a physical characteristic, it is called a genotype. This is what occurs with carriers. When it is expressed physically, it is called a phenotype. Female carriers are still susceptible to having bleeding problems, but for the most part, the healthy X chromosome is capable of producing enough clotting factors for bleeding to clot properly. In order for a female to be born with hemophilia, her father and mother must carry the hemophilia gene. This is not impossible, but is uncommon. This explains why hemophilia tends to be much more common in males. Although rare, hemophilia can also be acquired from a genetic mutation. In this case, a child could be born a hemophiliac, regardless of the fact that neither parent is a carrier. It is also possible for hemophilia to develop after birth. The blood may spontaneously produce antibody that blocks the clotting factors from working, leading to acquired hemophilia. People who are pregnant or suffer from an autoimmune disease, such as rheumatoid arthritis or cancer, are at higher risk to develop acquired hemophilia. But it's also known to emerge in people with no risk factors, making it a very unpredictable and therefore dangerous disorder. If it is not properly diagnosed and treated in time, acquired hemophilia can lead to serious bleeds, which could become fatal.
The video here will show the basis of ABO blood groups in humans.
In order to determine blood type, we will visually centrifuge these blood samples to separate the blood components. The ABO blood group is based on the presence or absence of two major proteins in red blood cell membranes, agglutinogen A and agglutinogen B. A person can have A and B, or neither A nor B in his or her blood cell membrane. These blood types are referred to as A, B, AB, and O. If a person has type A blood, his or her blood will contain antibodies against agglutinogen B. These antibodies are called agglutinins. Conversely, a type B person will have agglutinins against agglutinogen A. Blood compatibility can be predicted by understanding the differences between the donor and recipient blood types. The donor's blood generally only contains red blood cells with no plasma. Therefore, it is agglutinins in the recipient's blood plasma that determine if the donor's blood will be compatible. For these reasons, type O is known as the universal donor, while type AB is known as the universal recipient.
The DNA is deoxyribonucleic acid. It is located in the nuclei of cells which make up the body. Consequently, DNA can be considered as one of the building blocks of the body. To understand the exact structure and function of the body, it is essential to know what a cell is and how it is structured. The cell is the basic structure of the body. The human body is built of billions and trillions of cells. Cells of different organs vary according to their function. Each cell contains the hereditary material and can make copies of themselves by reproducing and multiplying. After a specific lifespan, the old cells die off. Part of the cell are called organelles. Human cells contain the following major parts. Nucleus. This is central part of the cell that carries the blueprint for the cell functioning and tells the cell when to grow, reproduce and die. It also houses DNA, deoxyribonucleic acid, mitochondria. These are the powerhouses of the cell and produce energy for the various activities of the cell. Cytoplasm. This is a jelly-like fluid within the cell in which the other organelles float. Endoplasmic reticulum, ER. This helps in processing the molecules, example, proteins created by the cell. Ribosome. These lie over the ER and process the genetic instructions or the blueprints within the DNA and create new proteins. These can also float freely in the cytoplasm. Lysosome and peroxisomes. These help in digesting foreign bacteria that invade the cell, rid the cell of toxic substances. Cell membrane. This is the outer lining of the cell. Within the nucleus, the DNA strands are tightly packed to form chromosomes. During the cell division, the chromosomes are visible. Humans normally have 23 pairs of chromosomes for a total of 46. 22 of these pairs, called autonomies, look the same in both males and females. Genes are hereditary material that lies within the cell nucleus. Genes, which are made up of DNA, act as instructions to make molecules called proteins. The DNA contains 4 chemical bases: adenine A, guanine G, cytosine C, thymine T. DNA base pairs. The DNA bases pair up with each other, A with T and C with G, to form units called base pairs. Each base is also attached to a sugar molecule and a phosphate molecule. Now, how does DNA replicate itself? The DNA can make copies of itself. Both the strands of the DNA open up and make a copy of each and become 2 DNA strands. Thus, each new DNA has one copy of the old DNA from where the copy is made. Mitochondrial DNA. The mitochondria contains small amount of DNA. This genetic material is known as mitochondrial DNA or mtDNA. The video here will teach us about the 2 stages of gene expressions: transcription and translation.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released. Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly-A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
The video here will show us some features of the Human Genome Project.
The Human Genome Project is a way of exploring our molecular selves. Almost all of our cells, the muscle cells that let us smile, the brain cells that perceive the humor in things, the cells of our eyes that take it all in, contain a complete set of all our genes, the genome. If we could journey inside ourselves into a cell, we would see 23 pairs of chromosomes packed into a nucleus. Each chromosome contains a long coil of DNA. If all the chromosomes were unwound, the DNA in just one of our cells would stretch 6 feet long. The DNA double helix contains 4 kinds of building blocks. An A always pairs with a T, a C with a G. DNA contains information to make every part of our bodies with its 4-letter language. Each of our thousands of genes codes for a specific part. RNA polymerase copies the information in a gene into a messenger molecule, messenger RNA. The building blocks of messenger RNA and DNA are called bases. The bases on one strand of the DNA specify the order of bases on the new strand of messenger RNA. The DNA always stays inside the nucleus, but messenger RNA travels out into the cytoplasm. There, a protein-making machine called a ribosome can read messenger RNA to make a particular protein. Every 3 bases of the messenger RNA molecule codes for an amino acid. Proteins are made of amino acids. tRNA molecules help translate the language of DNA and RNA into the language of proteins. tRNA molecules bring the right amino acids that the ribosome links together to make a protein. Proteins are the laborers. Some form structures like tendons and hair. Others perceive light, scents, and flavors, control chemical reactions, and carry messages between cells. To understand our molecular selves, scientists have read the 3 billion letters making up the DNA in the human genome. Different sets of genes interacting with complex environmental factors influence things like our looks, personalities, and risks for diseases like cancer and heart disease. A growing understanding of our genes and all they do will help us understand the complexity and the wonder of life.
Now in the end, let us summarize what we have learnt in this lecture. Genetics is the study of how genes bring about characteristics or traits in living things and how those characteristics are inherited. Augustinian monk Gregor Mendel developed the science of genetics. The different forms of a gene are called alleles. The law of segregation states that when an individual produces gametes, each gamete has only one of the two possible alleles from each parent, that is, the pairs of chromosomes 2n from each parent separate into two different gametes, one set of parental chromosomes per gamete 1n. The law of independent assortment states that different traits are inherited independently of one another. All the maternal chromosomes or all the paternal chromosomes do not end up in the same gamete, but rather the maternal and paternal pairs assort, separate into gametes independently of each other. Charles Darwin first conceived the idea of hereditary units when he published his theory of pangenesis in 1868. Construction of good quality microscopes led to the discovery of colored bodies in the cell nucleus that appeared to double and divide just before each division. They were called chromosomes. Hemophilia is a genetic disease caused to the inheritance of X-linked gene. It is said to be prevalent in the British royal family. DNA is deoxyribonucleic acid. It is located in the nuclei of cells which make up the body. The process of gene expression involves two main stages, transcription and translation. The Human Genome Project is a multi-billion-dollar project that aims to sequence the entire human genome for academic and research purposes.